Greetings, my name is Camilo Villaman. I'm a PhD candidate from Universidad Mayor, and I'm going to be talking about a new method developed to handle the problem of predicting CTCF binding using genomic and epigenomic features. CTCF is a multifunctional protein acting as a transcriptional activator, a repressor, an insulator protein blocking the communication between enhancers and promoters, and it's involved in the three-dimensional architecture of the genome. CTCF is essential on the establishment of topologically associating domains or TADs, self-interacting chromatin regions with increased intra-domain contacts, and CTCF loops with the help of cohesin, bringing together distant elements in the genome. Abnormal CTCF binding is related with disease and varies according to different cell types. Since CTCF binding is a problem that has not been completely resolved, we decided to generate a new method that uses genetic and epigenetic information to predict proper CTCF binding, with the idea of contributing new insights to the CTCF binding landscape. We used the following strategy for the predictor. We used FIMO and the CTCF weight matrix to find CTCF binding sites in the genome. Then we make windows of 500 pair bases upstream and downstream of the middle points of the predicted CTCF binding point, and then divide the windows into bins of 50 pair bases. Then we select different features including methylation, histone marks, and DNA accessibility, and we check if the feature overlaps with the beam. Each beam has two values. One is an intensity value and an average and an overlap value. The intensity value is the average intensity of the signal of the beam, and the overlap is how many bases of the feature are overlapping with the beam. We did this with each of the 4 selected cell lines, and we used 3 cell lines as a training set to train a random forest predictor, leaving the remaining one as a test set. In short, a random forest classifier is an ensemble of decision trees, a hierarchical structure built from the features of a dataset that infers conclusion about the value of an input according to the different observations of the dataset. The nodes are split based on selected subset of features. The final output of the random forest classifier is the mode of the result of every generated decision tree. We plotted ROC and PR curves to assess performance and plotted the area under the curve for comparison. The best performance was on the GM12878 cell line in terms of ROC AUC and PR AUC, and the worst one was HeLa. Here we can see the PR AUC of the 4 different trained cell lines used to train and then predict the value of the random forest classifier. And here we have the PR AUC of the same 4 cell lines. Then, to understand which features CTCF binding, we plotted the importance of each feature used. And as we can see, the most relevant feature is DNA accessibility, followed by methylation, and then followed by certain histone marks. And we could talk about H2AFC, which is the second most relevant mark after DNA and methylation. In conclusion, we built a CTCF predictor using random forest, and while the idea was to build a CTCF specific binding predictor, the ideas behind the predictor could be used to other DNA binding proteins, extending the functionality of this predictor. I also would like to thank the people that have supported me. This is the Network Biology Lab from Universidad Mayor, and this is the Chromatin Epigenetics and Neuroscience Laboratory from Universidad Mayor. This is our funding. Thank you for your time.
Thank you, Camilo. That was a really interesting talk. For everybody in the audience, if you have any questions, please post them in the Q&A, and Camilo could answer directly over there. In the meantime, I can ask a question. So, Camilo, when you mentioned that you tested on different cell lines, does it mean that you trained the model on the same cell line, or did you also try in the cross cell line prediction setting?
Yes, what I did was actually of the 4 cell lines, we picked 3 of the 4 selected cell lines, and we used those 3 as a training set to actually train the predictor. And then after training, we used the remaining last one as a test set. And after doing that cross-validation, we tested we ended up testing if we were able to actually predict if CTCF was binding in that cell line. And the results, we compared with the chip-sec values for CTCF, and then we calculated our performance values for that.
Okay, thank you.